The number is 136165. I'm going to talk about the effect of various abiotic factors and environmental extremes on microorganisms in terms of high temperature. Microorganisms that live in high temperature are collectively called thermophiles, which are facing the toughest environment in the world. They can survive 45 to 60 degrees, and hyperthermophile can even survive up to 122 degrees. They're usually found in deep-sea hydrothermal vents and hot springs, and most of them are archaea bacteria. High temperature means to the normal organisms is a degradation and denaturation of protein structure. Natural evolution has made thermophile gain the distinctive living strategies in the extreme environment. I will discuss adaptation to high temperature in terms of cytoplasmic membrane, cell wall, enzyme or protein, and DNA. Let's start with the cytoplasmic membrane. The saturated fatty acid in the cytoplasmic membrane make a strong hydrophobic environment, thus high stability. In hyperthermophiles, the lipid bilayer consists of long carbon chains with repeating units of isoprene side chains, and some of them have single layer of lipids, which are more stable. Besides, the lipid components in the thermophiles are able to change their shape as a response to temperature change. Chain length of the lipid acyl chain increases to limit the proton permeation rates and keep the membrane in a liquid crystalline state. Also, the tetraether lipid in the membrane make low IO permeability to keep sufficient PMF or SMF under the high temperature. Let's see the cell wall. The cell wall of thermophiles contains pseudopeptidoglycan, which can serve as function of peptidoglycan and make a basic structure of the cell wall. There are many charged residues, few uncharged polar residues which contribute to the thermal stability of the cells. In hyperthermophile, there is intra or intermolecular disulfide bridge in cell wall that can connect between proteins. Next is enzyme or protein. Hyperthermophile synthesis thermophilic enzyme that can function under extreme high temperature. Besides, chaperones in thermophile help to refold degenerated proteins. This interaction results in high stability of protein structure. The slow synthesized within the cytoplasm can also interact with protein and increase stability. Finally, the DNA of the thermophilic cell is chemically protected by divalent salts, which increase the stability of nucleic acid, and DNA is less likely to be depurated and hydrolyzed due to protection of potassium chloride and magnesium chloride. Furthermore, the smaller size of DNA leads to the synthesis of less diversity of protein family and make it a cost-minimizing mechanism.